Here's Andrea.
Hi, everyone. Welcome back. Okay. Awesome. Okay. Just before we get rolling with this next set of stuff, are there any burning questions from the morning session? We'll take your resounding silence to mean no. Okay. So the second sort of lecture in this series is, I've called it Human Genetics 1, and this is more historically motivated, I think, than it is any sort of understanding level motivated. But we're going to talk about a couple of concepts. So we're going to just quickly remember that we talked about meiosis this morning. And then we're going to get into definitions of haplotypes and linkage disequilibrium, which are ideas that are pretty central to modern genetic analysis. Then we're going to get into this idea of Mendelian inheritance. This is probably not something, as social scientists, you guys will use a whole lot, but I think it's very important to understand the history of genetics as a discipline and how these things began to get explored before DNA was ever sequenced, before we knew what DNA was. There's some fun variations on that, and then we will attempt to gently introduce complex traits and how they're sort of different from all the stuff we talk about today. So we talked about the human genome and its structure, that we have 22 autosomes, one pair of sex chromosomes, and a little tiny bit of mitochondrial DNA that is so small it doesn't even make it into this picture. We talked about meiosis and this idea that we have cell division, in which we have scrambling of the maternal and paternal genomic information, and then we have to divide the cells again so that we have haploid gametes, and we're not just raising our ploidy with every generation. This is a picture of that sort of shuffling or scrambling. And we talked about how, as this happens, as these cell divisions happen, that we can have changes, we can have mutations, we can have errors in the machinery that lead to all different kinds of germline mutations, and these are the ones that get passed on to our progeny. We talked about this idea that if there is a germline mutation in one parent, that it now becomes a variant in the population. So this is the new material for this hour. So a haplotype, this is a set of DNA variations or polymorphisms that tend to be inherited together. So you can think of them as a cluster. They're found on the same chromosome, usually pretty proximal to each other. And remember, we had this picture of recombination, and I mentioned that these crossover events or these recombination events occur at the same places or roughly the same places a lot of the time. So referred to as recombination hotspots. And the idea is that if there is, say, a recombination hotspot right here, that this end of this chromosome is going to likely be inherited together as a big chunk. And so we're going to see all the same variants on this red one and all the same variants on this blue one together, which is exactly what this slide says. So the idea that we have these recombination hotspots, we get big chunks of variation together, and as new mutations occur, they happen on a background that is already one of those chunks. So if we had a germline mutation, say in mom, that we might expect to see it occur on the red background, which is all the variation accumulated on that chromosome so far. That's sort of how haplotypes arise. This is the way we can think about it. So if this is sort of the reference sequence at a location, and then you have some non-consensus haplotype, so we have some level of variation, or some variation here and here, that if you have a new mutation on this first haplotype, you still have most of this consensus haplotype and then one different spot. And then similarly, for the unusual haplotype 2, you still have most of this black and blue one with one unusual variant there. And this is how folks construct things like phylogenetic trees and trace back our ancestry along chromosomes. So variants in close physical proximity, unless you're very, very near a recombination hotspot, do not get passed down independently. These variants on different chromosomes, however, or if you're sufficiently far away, they will, in general, get passed down independently. There are some strange exceptions to that, and I really do think those are exceptions, so we're not going to touch on those today. And then variants that are found together more often than you would expect it in independent segregation, we could say that they're in linkage disequilibrium, or that they're linked. And then variants that segregate independently, and I'll show you a picture of this in a second, are said to be in linkage equilibrium. And this is something that we as geneticists care about when we look at something like GWAS data pretty strongly. So we look at LD, this is just a screenshot from HaploView, and these correlation blocks. So you can think of this as the DNA from our samples up on the top, and these RSIDs, these are addresses of individual SNPs. So each one of these numbers is an individual SNP, and this is an address on it. And the red boxes indicate that there's a high degree of correlation between these SNPs pairwise. So every single box is a pairwise correlation. And as you can see, they kind of make these fun sort of pyramid shapes where you can see there's one block, and then the correlation dissipates. And then there's another block, and the correlation dissipates. And so this is known as LD structure. So the correlation between, I just said that. So the LD blocks are, different places will give you different estimates, but most of them tend to be less than 5 kb in length, but around 4 or 5. But they can be quite long. So this pattern of correlation, spatial correlation across SNPs, can extend to over 50 kilobases. The kilobase, kilo, 1,000 bases from this morning. So one thing that's very important to note here is that LD structure varies based on the population, and specifically, the size and age of the population can shorten the haplotype blocks. And so the idea being, I think the very classical example of this is in sub-Saharan Africa, the population is large and old. And so as you have many, many generations of a lot of people all existing through a long period of time, there's a lot more chance for recombination to happen. There's a lot more chance for recombination between different SNPs to recombine. Whereas something like Finland, which has gone through many bottlenecks in its history, so the population size got quite small many times very recently, those haplotypes tend to be a bit longer because they have had less time to recombine, sort of the intuition behind that. And then annoyingly, some recombination hotspot locations are different by population.
Why is that?
I'm not sure that it's completely known.
So, yeah, PRDM9 is a gene that controls like hotspot regulation. So there are sequences in the genome where you're more likely to have recombination events over time. And this PRDM9 gene regulates that sequence. It's a signaling variant, has this like particular domain that determines where binding actually happens in the genome. This genome is incredibly rapidly evolving. It's known as a speciation gene because it's changing really quickly over time. And because it's evolving so quickly, the binding regions where hotspots are likely to occur are changing. So because this particular gene that controls where hotspots happen evolves really quickly, that also means that where these hotspots occur is changing really quickly. So there are different hotspots in different populations at different frequencies.
So this is a picture of some differences over time. These are some reference panels from different ancestry groups. And you can see, as I said before, the Yorubans, which is the one sub-Saharan African population on this picture, shows smaller degrees of correlation. So we still have this very tight haplotype block here, but the rest of the pyramid is quite light in color, as opposed to many of these other groups where the correlation looks quite dense. Yeah?
I understand LD not really occurring between SNPs on different chromosomes as a silos fact, but why is that the case?
There isn't LD across chromosomes. So we're operating under the assumption that the chromosomes all line up and cross over independently. So what goes on chromosome 1 has no bearing on what goes on in chromosome 2. And so there won't be any spatial correlation between the two. Okay. So just a little bit of math here. So the coefficient of linkage disequilibrium, and some of this is historical math, you probably won't really use. But if we imagine that we have two alleles, we have an A allele and a B allele, we want to measure the difference between the frequency with which we see both the alleles together on the same haplotype. We'll actually use the laser pointer. And subtract the expected value. So how often do we see the A allele by itself? How often do we see the B allele by itself? And if they're segregating independently, we would expect this to be the probability that we see them together. And so we're taking this difference. So if this difference is significantly different from 0, we have now learned that these two things are likely linked. This, however, depends on the allele frequencies of this A allele and this B allele. So there were a couple standardizations proposed for this. This D prime is just this value standardized by its maximum possible value. So the idea being that if this is negative, we want to take the largest possible value in the negative direction, so the smallest possible value. And if it's positive, to take the reverse, and that's all this expression is saying. I haven't seen methodological uses for either of these measures. Most of the methods that I'm aware of use a correlation coefficient, which is this difference over the probabilities of each of the events independently, or more specifically, the squared version of this quantity. Because if we're talking about two locations, we don't necessarily care all the time that the T allele pairs with the C allele. We just want to know these two locations are very linked. And so this is probably the measurement that you'll be using, if at all. Okay. So with that introduction, we're going to rewind almost a couple hundred years here. And talk about how some of these ideas involving inheritance came about and how some of the earliest geneticists, even before the discovery of DNA, thought about these kinds of concepts. And a lot of this thinking informs how we think about genetic analysis now. So this is Gregor Mendel. He was an Augustinian friar, and he is posthumously recognized as the founder of modern genetics, though notice this date range is well before the discovery of the double helix and DNA. So Mendel's first experiments were on inheritance in plants. You've probably seen lots of pictures with Mendel and pea plants. So whether the peas are round or wrinkly and what color their flowers are. There were a lot of traits like that. And he designed controlled experiments to determine how certain traits were inherited from one generation to another. So he made things that look like this. You probably recognize some of this from some past schooling where you have plants with white flowers and plants with red flowers, and you cross them and see what the progeny look like. And so we're going to do some reviewing of that idea. So through his work, Mendel proposed a number of laws. We're going to talk about 3 of them. And some of these have some notable exceptions. But I'm presenting this from sort of a historical perspective, so we'll come back and look at these and figure out which pieces of this we want to use going forward and which pieces we maybe need to adjust. So the first law that he proposes is that if there are 2 different alleles at a locus, then one, the dominant allele, determines the organism's appearance, and the other one is recessive. So he's saying you have to have a dominant allele and a recessive allele. So a law of segregation. So this is something that came up in the discussion yesterday afternoon, this idea that alleles for heritable characteristics segregate during gamete formation, and they end up in separate gametes. So you have sort of 2 to choose from. And then the law of independent assortment. So inheritance of one allele, and so potentially a trait, is not dependent on another. So if your peas are wrinkly or smooth and what color the flowers are not correlated. And that's true for every trait, so says Mendel. And since I just talked to you about LD, we know this probably isn't true all the time, but there are going to be some cases where it is generally true. Okay, so it's hard to talk about inheritance without looking at pedigrees. I'm just going to give a quick primer on how pedigrees work. So squares are generally male individuals. The circles are generally female. And this line connecting two shapes tends to indicate a mated pair. The line descending from the mated pair is the progeny. In this case, it is a female progeny. And this sort of bracket-shaped thing with two of them indicates that you have siblings. So when we look at a pedigree, what we have is a pair of grandparents, 3 grandchildren, and then 2 of them, or 3 children, and then 6 grandchildren from 2 sets of parents. And when we look at these kinds of things, especially historically speaking, this shaded notation tends to indicate an affected individual. So if I had some trait that I was interested in, whether it was purple hair or the presence of a disease, you would indicate it with this shading. Okay. So a locus is a genomic location. Obviously, Mendel didn't know that. And it can consist of one or many base pairs. It's a kind of fuzzy genetics way of saying a location. A location can be very specific, like this building. A location can be very general, like Santa Barbara. And locus refers to both of those things. An allele is a different form or version. So at a SNP, we could say you have the A allele and the T allele, referring to which bases we have. But if it's a different type of variant, we could say there's the allele with 10 copies and there's the allele with 2 copies. Allele applies in both situations and it can get a bit confusing, but if we think of it as a version, I think that's usually easiest. Okay, so humans and mice, I imagine I'm going to show you an example with mice, are diploid, which means we have 2 possible alleles at both locus. So there are several classic patterns of inheritance, and we're gonna talk about a bunch of them. So if you imagine that I have mice living in one field, and then I go travel far away, and I go to a different field, and I take a mouse from each location and I cross them, and the mice from the first field have black fur, and the mice from the second field have brown fur, and I cross them to see what would happen, and all of the progeny have black fur, I might conclude that the black fur is dominant over the brown fur. And then I might assign this notation for the gene or the unit of inheritance as big B and little b. And so if this is a one locus sort of trait, I can infer that each one of these black mice is now heterozygous. So if I do this cross again, and I take 2 of these black mice from this first generation, and I cross them, I might end up with 3/4 black mice and 1/4 brown mice. And so this is how Mendel would sort through that. He would say, well, 1/4 of them, because these are independently segregating, 1/4 of them end up with the big B allele, the black fur allele. Half of them end up with 1 of each, but because black is dominant, you end up with black mice with that genotype. And then the ones with the 2 brown fur alleles end up as brown mice. There's also an experiment you can do called a back cross, where we take one of these guys from that first generation, you know he's heterozygous, and cross with a mouse that you believe to be in the recessive group, and you'll end up with sort of half and half. And this is how he did, Mendel in particular did a lot of checking, specifically, again, with plants, but this carries over to mice and flies. So in the mouse example, I said that the black coat color exhibits a dominant pattern of inheritance over the brown coat color, and the big B is the dominant allele compared to the little b. But if the allele of interest was the brown coat color, like if if that was the trait we decided to care about, we would say the trait is recessive. So dominance and recessive pretty much mean the same thing. It just depends on what your frame of reference is. So examples of dominant traits in humans, detached earlobes. So the idea that your earlobes hang free as opposed to attaching to your head. This tongue rolling thing, I'm sure everybody did that in high school at some point. And then there's some more serious ones like Huntington's disease, polycystic kidney disease. These tend to be very rare disorders, as dominant disorders tend to be. And so we see this in human beings, just as we saw it in pea plants and in mice. So if this was our pedigree in humans, you can imagine a dominant inheritance pattern might look something like this, where many carriers of dominant traits, especially dominant diseases, tend to be heterozygous. And so the offspring have a 1/2 chance of inheriting that allele. And when you inherit the big B allele, you are affected with the trait. So it's like fairly simple and fairly deterministic. So recessive traits, as I said before, this is pretty much the same thing as dominance. It's just the brown fur is the trait that we're saying is our trait. That's the thing we care about. And so we get the same results as before. And we would end up concluding that the trait itself is recessive. So we would have this sort of pedigree where only the folks with the 2 little b's end up showing the trait, and everybody else, as long as they have at least 1 big B allele, would be unaffected by our trait. This is fairly simple and fairly deterministic still. So, as I said before, these 2 words really mean the same thing. It just depends on which thing you call the trait of interest. This is especially relevant for disease, because if you're going to talk about inheritance of a disease, usually the disease state is the trait of interest. So we had some examples of dominant diseases. There are a lot more recessive diseases. Fragile X syndrome is definitely in this category. Color blindness, Duchenne muscular dystrophy follows this pattern, as does hemophilia A. There are several different types of hemophilia. Mendel also coined a bunch of other terms that are less useful today, but I'll take you through a couple of them. So codominance is this idea that there are alleles that sort of end in a tie. They're both expressed, and they're both fully expressed. So if I have this white cow and this black cow, and their progeny end up being spotted cows, we have cows that are both white and black. Something like incomplete dominance would mean that you end up in something of a compromise. So if you have these dark purple flowers and these white flowers, but the plants are the same, when you cross them with pollen from one to the other, you end up with something in between, like a lavender, like a light purple flower. And that would be incomplete dominance. These were sort of considered weird exceptions, and we now sort of think of most things this way. So we talked about the sex chromosomes this morning a little bit, and how they have slightly different rules. And so, as we talked about this morning, females have an XX karyotype, males have an XY, and then the mitochondria, which I didn't tell you this morning, are maternally inherited. So your mitochondria always come from mother. The paternal men do have mitochondria, but they're never inherited from father to child. So if we had something on the mitochondria, and mitochondrial genomes are variable enough that we can usually tell them apart. So this person, this mother, gives her mitochondria to all of their children. This mother gives all of their mitochondria to all of their children. And this mother gives all of their mitochondria to all of their children. So if something follows the maternal line very strictly, it is possible that it could be mitochondrial. Mitochondrial disorders do exist, but they're incredibly rare. Mitochondria are fairly primitive and fairly important to cell function, and so most terrible dysfunction with them isn't really compatible with life. All right, so similarly, if something follows the father's line but only affects the male offspring, you might suspect that it's on the Y chromosome. So if you have something going on the Y chromosome, it would be passed very deterministically from fathers to their sons. And a mother would have no way to transmit it because they don't have a Y chromosome. I should mention there are not very many genes on the Y, so this also doesn't happen super often. X-linked dominant things, so this is the trait of interest following one X chromosome, but remember that we have a random draw of which X chromosome we get from mom, because they have two. And so these two offspring might have ended up with the red X from mom, but this offspring could still avoid it, because you still have a half chance of drawing these. And then similarly, this person has two X chromosomes to pass on, and so they could pass on the red one or not. And lastly, this is slightly more common, is the X-linked but recessive. So the way this ends up, because X and Y are a little bit weird, mother has two copies of X, but father only has one. And so if something's recessive but you only have one copy of X, you don't have the other copy to balance it out. And so males with an affected X chromosome will still end up being affected. And that's why for many of these diseases, they show up with much higher prevalence in males. So you'll see a male affected, male affected, male affected. And the only way you will ever see a female affected is if they had an affected father and inherited the affected allele from their mother. Okay, so nowadays, this is fast forward to present day, we talk about traits as being Mendelian when we know the individual's allele is at a single locus and it follows a mode of inheritance that's as simple and deterministic as this. So these are all the things we just talked about and their names. I think the simplicity and the deterministicness, and there's a couple adjustments we can make to that, but it's largely deterministic. So we're going to re-examine Mendel's laws here. So the law of dominance, we know that there are a lot of traits that don't actually meet this criteria at all. However, he did find a lot of them. The idea that alleles for a heritable characteristic end up in different gametes, that is true. And that is true no matter how polygenic you are. And then the law of independent assortment, here is the inheritance of one allele not being dependent on the other. And this is true as long as you're not proximal, right? If you're on two different chromosomes, it's fine. But this is not true when you're an LD. And that's probably why I started. All right. So long before the discovery of DNA, Mendel was already outlining these principles. He studied peas. He really did seem to restrict himself to traits that were on different chromosomes, and it's up to the interpretation of the reader to decide if that was on purpose after the fact or if he just was lucky that way. But we do know that some genes have proximal relationships, and in this century, there were quite a few studies talking about traits that are linked, and so we're going to define linked traits. So this is an example using fruit flies. I'm looking at two traits right now. So I'm looking at the color of the eyes. These guys have red eyes, and these guys have white eyes, which is shown as sort of a purple color in this cartoon. But if you actually look at these flies under a microscope, their eyes is sort of eerie, translucent white. And the flies also have long wings, and short wings. And so similar to the mice in the field, if I take fruit flies from one basket of fruit in one building, and they all look like this, and then I take fruit flies from a totally different location and they all look like this, I might cross them to see how those two traits look. And if all the offspring have red eyes and long wings, I might determine that red eyes are dominant over white eyes, and the long wings are dominant over the short wings. Was there a question? No? Okay. So I might assign those alleles some letters. We have E for eyes, and the capital letter is going to be the dominant one, and W for wings, and again, the capital letter will be the long wings, and the small w will be the short wings. And so from this cross, using Mendel's laws, I can infer that all the offspring have this kind of genotype, so one capital letter and one small letter, and now I can't read that as anything but U, which I think is why there's giggling. So we do this sort of back cross experiment that we talked about with the mice. We have this first generation crossed fly that we know is heterozygous with this one that has both of the recessive traits. And you can imagine that we have 4 possible combinations we might come up with. We could have the one that looks just like that one. We could have a fly that has short wings but red eyes. We could have a fly that has long wings and white eyes, or we could have a fly that looks exactly like this parent. And if these 2 traits were on different chromosomes, we could expect the rate at which we would expect that to happen. And if we think about this as a likelihood, very simple likelihood, you know that each progeny has 1/2 chance of inheriting the big letter allele from the parent that is heterozygous. And we really don't have any randomness in this fly, right? Because they're homozygous for something that's recessive. And so you could easily compute that we would expect to see a quarter, so that all the populations of these flies would now be the same. However, if we do that, and our observed data, let's say we did this on 1,000 flies, end up looking really different, we might think something's up. And so if I expect to see a quarter of the flies in each of these bins, but I see the vast majority of these flies, I put the numbers in the wrong place. This one is supposed to be here. The idea being that if we saw all flies that look like this one, and like this one, and fewer flies that look like this one, that we might think the traits are linked. So if you visualize the chromosomes, we might think, okay, we have the capital E and the capital W together, and the small e and small w together. And that is also wrong. I think I just put the flies in the wrong place. These 2 are supposed to switch. And I will switch it for your PDFs as soon as we're done. So if we observe this, we can say these 2 little ones, so we expected by random to see a lot more recombinants than we actually saw. So we could say out of 1,000 flies, how many recombinations did I see? I'd say, okay, I saw this rate of recombination. And this actually is how we end up defining the distance measure centimorgan. There's a whole history about why it's called centimorgan that I'm not going to get into right now. So if the technology were available, we could just count the bases between one allele and another. 1, 2, 3, 4, until we get there. But prior to the availability of base level sequence data, genetic distance was measured in recombination. So if you cross things, how many recombinations do you see? So a centimorgan is defined as the distance on a chromosome where the expected number of crossover events is 0.01. So the idea here is that if you did 100 of them, you would see 1 recombination. So this is 1 per 100. And on average, a centimorgan corresponds to about 1 million base pairs, but again, we talked about LD, right? And so LD makes this distribution very lumpy and very complicated. But occasionally in genetics papers, you will still see things referred to in centimorgan. So this idea of centimorgans is still interesting in the history of modern genetics because we have this idea of microsatellite tandem repeats. So this is a type of genetic variation. We talked about structural variation this morning. We talked about CNVs. This idea that there are repeated regions of the genome where you might have many, many copies of the same sequence, and you might have fewer copies. And so these are highly variable and allows tracing of parental origin pretty easily. These were often measured by fluorescence. This is an older kind of buggier technology that is not used very much anymore, but this was all the rage maybe 20 years ago. And so these analyses were known as linkage analysis. If you could get these genotypes and a trait that you were interested in, now you could do the same thing I did with the flies, with the eyes and the wings, except one of my traits is now the number of repeats. So on each of these people in my pedigree, we have the number of repeats. And because we have 2 copies of every chromosome, we have 2 numbers of repeats. So we have a 1 and a 3, and a 2 and a 10. And suppose I'm a researcher in my disease of interest, and I'm pretty sure that longer repeats are going to be associated with my trait. So I'm going to say, I think the 10 and my trait are linked, or that's the thing I want to test. And so it is possible, if you make some assumptions about how far they are apart, you can test the null hypothesis, these things are linked, and these things are not linked.
So
Yeah.
when you say linked, are you talking about so I think you used linked to talk about the gene being linked to the phenotype.
Mm-hmm.
But it's not about the genes being linked to, I guess it's not 2 traits here. Okay.
Yeah, yeah. But I think that's worth clarifying. So the idea is that if this is my chromosome, and that's the middle, that if here's my repeat, and I have a vague idea of where this repeat is, and I think that the locus that causes this disease, it's not going to be in this repeat. The repeats just generally aren't that interesting. But I'm saying I'm going to rely on LD to say, well, I think it's nearby. And so if I think my trait lives here, then the number that we get from this repeat and the trait are the 2 things we're linking. And so this is a pretty short little pedigree. There's not a ton of information in this. And if we said, if these are the genotypes we got back, and we ended up with that one, we could construct a likelihood ratio. This is known as a LOD score. It's supposed to stand for logarithm of odds, but you might realize that that acronym doesn't work out super well. I think LOD score just sounded good, and that's what folks decided to use. I have not found an acronym for it that makes sense. But this idea that you take the log base 10 of the ratio of the likelihoods. And so I made an assumption about how far they are apart, and all I'm doing is saying, okay, under the null, they would be segregating independently. So the odds of, or the probability of seeing each of the things that I saw is 1/2, specifically the phenotype values. And the probabilities, we had 2 non-recombinant offspring and 1 recombinant. So we would end up with a LOD score of 0.07. What does this mean? Similar to many things in genetics, there was sort of an agreed upon threshold, and folks decided that a LOD score of 3 or higher was significant. That means that the likelihood of your alternative outweighs the null by a factor of 10 to the 3. That was sort of a standard thing for a while. Obviously, this is not significant. However, if it turned out that later in life this individual became affected, our LOD score would go up quite a bit. And a lot of very simple traits were mapped in this way. using these VNTR variants. They were mapped to rough locations on chromosomes using this kind of analysis. You can combine information across multiple pedigrees and so on. So we mentioned LOD scores of 3 are considered significant. To get a significant LOD score, you often needed a pretty large pedigree or many pedigrees. And of course, if you're drawing from different family samples, you might imagine that a different allele is associated with the disease, and then it gets quite hairy. So linkage models can be expanded to include effects for sex, age, and penetrance. Penetrance is this idea, so we're taking away the certainty. We're taking away this idea that it has to be prescriptive, that if you have this genotype, you must have this phenotype. And we allow there to be a little bit of probability in there. And so the probability that you are affected given a disease genotype would be called adding the penetrance parameter. All right. So, wow, we're moving really, really quickly. Do you guys have any questions? No? Okay. So I know you all know what a phenotype is, but I like having my definitions be complete. So the phenotype is an observable characteristic of an organism. So phenotypes result from the genetic composition, the environment, and probably some interaction between the two. And this is not something that we understand super well. So phenotypes can be Mendelian inherited. That means they involve a single locus, and they involve many loci. That they can also involve many, many loci and have an inheritance pattern that's super complicated. And so complex trace genetics provides a new toolbox for addressing non-Mendelian traits. And that's what I was hoping to talk about next time, but we are super early. So I'm going to stop here and ask you guys if you have any questions about this stuff or the stuff we talked about in the morning.
Can you repeat the number of copies each interval there? I understand it's the refinement of the Mendelian slide. I didn't find anything.
Is that this slide?
Maybe the one before that. Yeah.
Okay. So you're talking about the meaning of these numbers.
Yeah.
Here? Okay. So we talked this morning about this idea that there are structural variants, right? That there is, maybe if this is my chromosome, that there might be a region here that when I duplicate, I get like 3 of them, because this region is repetitive and my replication machinery has somehow slipped and just done this bit a bunch of times. Those kinds of mutations are pretty variable. They have a higher mutation rate than something like SNPs, but they still can be passed down in a way. And so there are some technologies that will measure how many copies of this sequence do you have. And so that's what these numbers mean.
So
Here.
they're easier to measure than, like, why were they measuring those instead of?
They were, this is pre-SNP technology. So yeah, they were, it was a PCR amplification step, I think. If anybody is a molecular biology person and actually did this in grad school, stop me. This was on its way out as GWAS was rising. My impression is that it's a little bit more one at a time, that multiplexing them is a bit hard, and that telling the difference between, like our conversation with CNVs, telling the difference between 1 and 2 is not that hard. Telling the difference between 3 and 4 is a little harder, and telling the difference between 10 and 12 is very, very difficult. And so it has that a little bit logarithmic attenuation. And so these had to be selected very carefully so that they were appropriately variable. Yeah.
Actually add to a little bit of that.
Yeah.
My understanding is that it's used in police forensics, these kind of repeats, right? So it's still sort of continues to exist in that work. Not exactly the linkage analysis, but sort of the use of those tandem repeats is how forensics are done.
That's cool.
Yeah.
Yeah.
Was it where they like pass the electricity through the thing, like the jet? Is that? Never mind. This is something. Is it electric? Yeah, is that what it is? I don't know. Okay. When you said forensic, I'm like, oh yeah, I think I remember pictures of. Doesn't matter.
I think these days in forensic, though, they're moving toward doing
Yeah.
genome-wide data.
No, you're right. Actually, there's some papers which found that the SNPs highly coexist with the tandem repeats. Or with
Mhm. Yeah, go ahead.
Is the penetrance stuff, is that explained by like gene-gene interaction or some other, like is the way to think about that is like non-additive?
Yeah, I think the way to think about the penetrance is more of a stochastic component to this very deterministic model.
So it could still be additive and
It could still be additive, and I think the penetrance parameter is more of a statistical adjustment so that the data can actually be fit by something other than a recombination, which is highly unlikely when things are very close together. Remember, the person I learned this from was a geneticist named Nancy Cox, and she gave us a pedigree, and we all said, well, this is autosomal dominant. It was a pedigree of being a physician. And she said, this is autosomal dominant with reduced penetrance in females. And there was a pedigree of being an MD. And so that's how I remember penetrance as that stochastic component. And there could be any number of reasons for that. Like if you are a girl, you're less likely to be an MD, even though that trait has nothing to do with genetics.
Or does it?
Everything's genetic, right. But yeah. Yeah.
Concerning copy number variants, what are the implications of having duplicate, and especially for transcription? Because I guess that it will depend whether this copy number variant is an intron or an exon, right?
Yeah, and in reality, they tend to be neither. They tend to be in intergenic regions. So we talked this morning about things being in exons and introns, but in actuality, you might have a chromosome that's very long, and you might have a gene here with some little exons, and then a vast expanse of chromatin that encodes for nothing. For a long time, the popular literature would report this as junk DNA, the idea that it doesn't do anything, we just carry it around with us from generation to generation. Newer research, I think the jury is still out on that. There's quite a bit of positing that these things have structural roles in the integrity of the DNA and how things hang together, whether or not these things end up in the right places when it's time for them to be transcribed, and those sorts of things.
Do they have any association with phenotypes, like having larger number of copy number variants?
Yeah, so there are a couple of such examples. Fragile X is an example like that, where there's a lot of copies of the same repeat on the X chromosome, and what ends up happening is there's so many of them, the chromosome gets a bit fragile and breaks. So they can be associated with phenotypes in a causal way, like that one, but more often, like in the case that I'm presenting here, they're merely tagging along. They're LD friends with something that's actually functional. And so like if there's LD between these two loci, then this signal, the association that I'm getting from this, has nothing to do with causation and everything to do with its proximal relationship to this gene here. Yeah.
Is there a threshold for the coefficient for LD? So do you know what determines high LD versus moderate LD?
Yeah, so LD, you're bounded if we use the R-squared measurement, which is my favorite one. A lot of these. Okay, so this is a difference in probabilities or frequencies, and this is a function of those frequencies. So LD is bounded between 0 and 1. Folks that do LD score, maybe bail me out if I get this wrong. I think it can be thought of as a correlation pretty strongly. The idea that either things, they track together or they track separately. They track independently. And so two things that are inherited together all the time, my dissertation advisor used to call them LD buddies. So two things that are just inseparable. if you have A here, you have T here, and this does happen, that your LD is 1, meaning they always track together, and then something that is completely independently inherited is got an LD of 0.
Sorry, about this, I was wondering whether on the, let's say, mechanism level, right? So you can think about with 2 genes that are related and they are come together in the LD, but then is it used also to understand what's the mechanism behind? Or not really? I don't know,
So,
logical. So let's say these 2 genes are both related to the production of certain protein, right? And then from that you can think this is associated with a certain phenotype.
I see. Okay, there's a couple things in there. So one thing is that if you have 2 genes and they're right next to each other, it does happen sometimes. The MHC or the HLA, which is involved in immune function, is a great example where the 2 genes might have related functions. But very often their functions are quite different and have very little to do with each other. And so in general, say I have an association with a marker that's here, and it's the LD block easily includes these 2 genes. I see that as more of a problem of deciding which gene is more interesting or if it's both of them, rather than saying, well, they both must be interesting, because LD masks our ability to tell those things apart. So it's more of an identification issue than it is additional information. Cool. Okay. I guess if there's nothing else, is it okay if we have a slightly longer break this time, or?
Yeah.
Okay.
Absolutely. So.